Osmosis is a process in which molecules of a solvent pass through a semi-permeable membrane from a less concentrated solution into a more concentrated one, thereby equalizing the concentrations on each side of the membrane. In order to understand osmosis, we need to understand diffusion in general. Diffusion is the net passive movement of particles or molecules from regions where they're more highly concentrated to regions where they are less concentrated. In other words, there is a concentration gradient. The particles are said to move down the concentration gradient. It's important to notice that this is a passive process. There's no energy being put into the system to do this. It happens because the particles are always in a state of random motion, and in accordance with the second law of thermodynamics, the system will always trend towards increased entropy. Diffusion increases the entropy or disorder in the system as different substances mix together. Diffusion will cause the net movement of particles down their concentration gradient until the gradient no longer exists. If you wrap a membrane around the area of highly concentrated solute, you could still have passive diffusion, as long as the membrane is permeable to the solute. In fact, you could still have passive diffusion even if the membrane is not permeable to the solute, but that's called facilitated passive diffusion, and we'll come back to that in a moment. So, whereas diffusion refers to the movement of molecules from an area of high concentration to an area of lower concentration, what happens if the molecules can't move across the membrane? Well, nothing really, unless the membrane is impermeable to the solute, but permeable to the solvent. In other words, the particles can't move across the membrane, but the water can. This is osmosis, and osmosis is a special case of diffusion. Membranes that have this property of being permeable to one thing and not another are actually the norm in biology. These are the so-called semi-permeable membranes. So osmosis is the movement of a solvent, in biology that's usually water, across a semi-permeable membrane toward a higher concentration of solute. So remember that passive transport relies on the second law of thermodynamics to drive the movement of substances down the concentration gradient, sometimes across cell membranes. And as a little throwback to the flight physiology course that some of you may have done, we could also say that these substances are following Fick's first law. So, can you have a molecule passively diffuse across a membrane even when the membrane is impermeable to the molecule? Actually, you can. You just need to punch holes in the membrane. This is actually a common feature in real-world cell membranes, which is why it gets a special mention here. The so-called holes are usually in the form of a protein channel embedded in the lipid bilayer, the cell membrane. What makes them interesting isn't so much that they exist and that they make the cell membranes leaky to certain molecules, although that is interesting because you can have things pass through a membrane that otherwise wouldn't be able to. But what makes it really interesting is what you can do with the protein channel. One of the main things you can do with it is add a gate. You can actually choose when the channel is open and when it's closed. Now, opening and closing the channel may be an active or energy-consuming process, but the diffusion itself is still passive. What about the case where you want to move a molecule across a membrane against its concentration gradient? This is called active transport. In this case, we want to move the little yellow diamonds into the cell, but there's already a high concentration of little yellow diamonds in the cell. We can use the cell's power source, molecules called ATP or adenosine triphosphate, to actively change the shape of a protein channel and effectively pump the diamonds into the cell's interior. ATP molecules are the batteries of metabolism. When ATP is reduced to ADP, it releases a little bit of energy, and the energy is used to change the shape of a protein embedded in the lipid bilayer that effectively becomes a molecular pump. Incidentally, recharging the battery, or converting ADP back to ATP, is an oxidation reaction, and it's literally one of the reasons that we breathe. Just to confuse things a little bit, remember that there are not just concentration gradients, there are also electrical gradients. If I concentrate a large number of charged particles on one side of the membrane, whether with or against our concentration gradient, it doesn't matter, I will have created an electrical gradient. In much the same way that the concentration gradient causes particles to move across the membrane, the electrical gradient can do the same. These electrical gradients are crucial to nervous impulses, muscle contractions, and cardiac physiology.